So we are going to cover some, these next couple of classes are going to be more heavily reliant on kind of research article based evidence for mechanisms responsible for the control of ventilation and the transition from rest to exercise, and then also the concept of respiratory limitations to exercise. Okay, so we'll be providing some evidence ultimately for this concept of a respiratory muscle steal phenomenon, where the high work of breathing that's incurred during exercise has the potential actually to kind of strip blood away from the peripheral locomotor muscles. So those are kind of our two major outcomes of the next couple lectures. So as a review from last class, we ended and we talked about how the ventilatory response to a step increase in exercise intensity going from rest to exercise is very rapid and immediate. It's characterized by this kind of three-phase response, where you have this phase one, a very rapid and intensity-dependent increase in ventilation. You've got this transitionary period, or this kinetic period, where ventilation is then adapting to the increased metabolic demands, and then ultimately, at least during a light and moderate intensity exercise, you get to a steady state where ventilation and metabolic demands are matched. But during heavy intensity exercise, above the anaerobic or ventilatory or lactic acid threshold, we know that we have to have a ventilatory compensation for that metabolic acidosis, and therefore ventilation doesn't reach a steady state condition where ventilation is matched to metabolic demands, because you need to ventilate in excess of metabolic demands to compensate for those high rates of glycolytic flux and the metabolites that are being generated as a result, to try to minimize the extent of the acid-base balance disturbances. So we highlighted last time that these phase one response, so that very much that within breath immediate increase or breath to breath increase in ventilation is intensity dependent. And so we talked about some of the potential candidates that could exist in both the feed forward and feedback control mechanisms of the respiratory control system. And so we talked about central command, which again is areas in the higher brain centers that can send efferent signals to the respiratory and locomotor muscles that can control ventilation, say independent of the respiratory pattern generator. And so again, we said, for example, immediately right now you can hold your breath or you can voluntarily hyperventilate. And the idea is kind of a classic example of how you have higher brain control of your respiratory muscles. And that's something that can occur very, very quickly. So central command is something that could explain an immediate increase in ventilation at the onset of exercise. And somebody came up and asked me a question at the end of class, and basically the idea was that even in anticipation of exercise, if you bring somebody in the lab and you say in 3, 2, 1 you're going to start exercising, even at say 3 seconds or when you're starting the countdown, their ventilation will rise in anticipation. They haven't done anything. There's no metabolic change, they have no proprioceptive feedback, there's obviously no metabolic disturbance, but their ventilation will rise in anticipation. There's not data that I'm going to show you here, but there's some really interesting studies that were done, I believe it was from Oxford, where they looked at individuals that were hypnotized. So they did an exercise test, a step increase in exercise in reality, and measured the ventilatory response. And then what they did is they hypnotized individuals, and then again, they had them all measuring on their, on breathing systems. They hypnotized the individuals and they asked them to visualize or think about the exact same exercise intensity that they did. And without any physical exercise, just imagining the exercise, the ventilation changed almost exactly like it did when they were exercising. So again, exercise itself is not necessarily obligatory to drive ventilation. You can have the idea of these higher brain centers, which could provide a very fast feed-forward control of ventilation. We also talked about neural input from type 3 and type 4 sensory afferents located in the peripheral muscles, as well as the respiratory muscles, as well as in the vasculature, the joints, the tendons, and things like that. And so really, the mechanoreceptors in particular are probably the candidate afferents that are likely responsible, as you'll see, for providing immediate feedback-related information about exercise. So as soon as we start moving, as soon as we start pedaling on a resistance, there's an immediate change in the proprioceptive information that would be sent back to the brain, which would signal basically a compensatory increase in ventilation. Basically the leg and the joint saying, hey, you're doing more, so now increase ventilation to try to support that anticipated gas exchange. So while that is an afferent feedback, that's a nerve that comes from the periphery, it's such a fast response that we kind of loop it into the feed-forward mechanism. So it's a fast neural feedback, but really it operates almost as quickly as a feed-forward mechanism, because it's not really responding to an error per se, right? It's not really trying to correct to an error. It's not like a feedback, classical feedback loop, but it is a feedback-mediated mechanism nonetheless. We talked about the metaboreceptors. So again, metaboreceptors, kind of their name is that there requires a metabolic disturbance. Neither your air conditioner nor your furnace turn on when it's the appropriate temperature in the house. It either needs to be too hot or too cold in order to signal an error. So at rest and at that immediate onset of exercise, there's probably not much in the way of a metabolic disturbance, either systemically or at the level of the muscle tissue, that could explain that immediate increase in ventilation. And then finally, we have the central and peripheral or carotid chemoreceptors. And so these are really predicated on the ideas that you would have some form of systemic hypoxia, acidosis, or central acidosis that would stimulate these receptors. And again, this is not necessarily the case at rest. It may be the case during very heavy intensity exercise when we have this systemic metabolic acidosis and these receptors may become active. But again, focusing most importantly on kind of that immediate initial increase in ventilation, probably not major candidate drivers of that ventilatory response. So what we'll do now is we'll go through some experimental evidence and support for and against the role of these various these feedback and feed-forward mechanisms. So about, what is this, about 35 years ago, this is kind of the classic experiment published again, about 35 years ago, where they looked at the role of central command in terms of driving, in terms of trying to drive ventilation. And so I'll give you an idea of the experiment here. Here what we have is we've got this cat, and this is I don't know why it's cut off today, but this is as you see on your slides, this is a cat with basically its cortex removed, its vagus and carotid nerves cut. Okay, so basically, this does not have any vagal feedback coming from the periphery and the lungs. So it doesn't have any stretch-sensitive, say, mediated receptor feedback coming from the lungs. It also doesn't have functioning carotid chemoreceptors, so those nerves are cut as well. Then it doesn't have a motor cortex, so basically it was removed. Okay? So what you have is you've basically got this cat that is not anesthetized, but doesn't have key feedback mechanisms and also doesn't have a cortex. It's been surgically ablated. So what they do is that in this experiment, they drop an electrode into the diencephalon, part of the brain that they believe was, maybe responsible for central command. Then they measure things like carotid artery pressure. They put a needle into the phrenic nerve. So the phrenic nerve is the major nerve that innervates the diaphragm, which is the major muscle of inspiration. And so an increase in phrenic nerve activity is the neural equivalent of ventilation. It's basically the best measure, one of the best measures of the drive from the brain to the muscles to support ventilation. Okay? Then we have end-tidal CO2 levels, so a measurement of how much carbon dioxide, so a measure of the systemic chemostimulation that would exist. Then they got EMG activity of the peripheral muscles here, and then they also had temperature, rectal temperature. So they basically were trying to look at, I believe they were able to maintain temperature of these cats as well, to prevent a temperature-induced driving of ventilation. So what they did is that in this particular cat, under this thing, they suspended it in a sling because basically it's brain dead, but it's not paralyzed. Okay? So they stick this cat in a sling and they put it on a treadmill, and then what they do is that they begin to stimulate the diencephalon. So they start putting an electrical pulse into the diencephalon to stimulate brain activity or to stimulate central command. And all of a sudden this cat starts walking. It's not actually walking, but it's walking on the treadmill. So what you can see is that when they induced locomotion by stimulating the diencephalon with this electrode, what you can see is that arterial blood pressure rose. So again, this is what we would expect. Blood pressure rises. So central command, I believe this was talked about by Charlotte. Central command is a driving factor that can increase mean arterial pressure in exercise. If you look, quadriceps EMG increased, so the muscle's moving, right? It's active, both like proprioceptively, mechanically, and perhaps also a little bit metabolically. And you can see that with the increase in physical activity and the induced locomotion, phrenic nerve EMG increases. So this animal basically starts to ventilate. And you can see that they're hyperventilating because actually the airway CO2 levels decrease. So now they get a stimulation of ventilation that blows off CO2 in an animal that has no carotid, no vagus nerves, and its cortex removed. Just stimulating the diencephalon. Okay? So now the problem with the interpretation of this is that, is this a movement-related phenomenon? Is this a chemoreflex-mediated phenomenon? Is this a central command-mediated phenomenon? Because in this example, yeah, while you maybe don't have feedback from the vagus nerve and the carotid nerve, and and they don't have a cortex, is that you still have movement of limbs and joints. So you got proprioceptive information from mechanoreceptors. You could have metabolic stress coming from the respiratory or limb muscles, right? Because it is moving. So you could have feedback from those type 3 and 4 sensory afferents. And you could also have activation from the muscles themselves. So basically, if you would want to, in a reductionist approach, to get down to kind of the central command, you would want to eliminate movement of the muscles so that they're not metabolically active and they're not moving, so you take away basically the type 3 and 4 afferent feedback. So what you're left with is presumably the central command, right? Temperature-controlled animal, no carotid nerve, no vagus nerve, no cortex, no type 3 and 4 muscle activation. So what you can see is that they basically then what they do is they have this cat effectively paralyzed then. So they infuse it with, I believe in this case it was like a curare, like a neuromuscular blocker, so that electrical activation of the brain doesn't actually translate into muscular activity. So the animal's effectively paralyzed. And so what you can see is that going from rest and then into fictive locomotion, you can see that even though you can see that actually there's bursts. So when they begin to stimulate the electrode, you can actually see that the femoral, the biceps femoris nerve in the right and the left leg are active, but it's not actually translating into movement, right? Because the animal's paralyzed. So basically the nerve is firing, but it's not actually translating to muscular contraction. And you can see here that actually the phrenic nerve activity increased. So no feedback from carotid chemoreceptors, vagus nerves, no cortex, no movement, no activation from type 3 and 4 afferents, but stimulating the diencephalon in a paralyzed animal increases phrenic nerve activity. So even in the absence of changes in ventilation, because this animal didn't actually increase its level of ventilation, even in the absence of changes in ventilation, because I mean you can see here that the end-tidal CO2 levels didn't change, right? So the animal didn't ventilate and decline their end-tidal CO2 levels like they did, so it didn't actually hyperventilate. So in the absence of change in ventilation, tidal volume, breathing frequency, feedback-related information from the lungs, the carotid chemoreceptors, as well as from type 3 and 4 afferents, stimulation of the diencephalon increased phrenic nerve activity. So the idea here is that in the absence of all other feedback-related mechanisms, activation of the brain can drive ventilation. Is that, you see it, that's pretty reasonable? Yep.
Can you repeat that?
Pardon me?
Can you repeat that last one?
Yeah, so the idea is that even in the absence of any feedback-related mechanisms, activation of central command can drive ventilation. So, you can see here on the left that electrical stimulation of the hypothalamus, i.e. activation of central command, stimulated both locomotion and respiration. But in the absence, or in the paralyzed cat, phrenic nerve activity increased during electrical stimulation of the hypothalamus, even in the absence of respiratory and peripheral locomotor muscle contraction. So again, this is kind of the classic experimental evidence that central command can likely contribute to that immediate increase in ventilation at the onset of exercise. I do not know why this is so shifted, but further evidence for central command actually, and remember we said that the phase 1 response was intensity dependent. So that's an interesting phenomenon too, is like how does an individual, question? Sorry? Yeah. So the question is that how, what's going on? Oh, I can't change that. So if you actually look at the effect of grading or ramping up the electrical activation of that diencephalon stimulating electrode, which would be an idea of increasing the amount of central command output. Okay? So here on this plot you can see the mean stimulus current on the x-axis and the phrenic nerve activity here and the femoris nerve activity here. So we're looking at the red and then we're looking at the blue for respiration. And so electrical stimulation of the hypothalamus in these paralyzed cats, again, so this is in that paralysis condition, resulted in parallel and intensity-dependent increases in both respiration and locomotor activity. So even in the absence of muscular contraction and ventilation, increasing central command in an intensity-dependent manner resulted in intensity-dependent increases in the attempt to activate the locomotor and respiratory muscles. So the idea here is that even when the muscles are able to contract, presumably that's occurring as well. So thus, central command likely increases as a function of exercise intensity with attendant or accompanying and proportional increases in respiration and muscular contraction. And this would make sense. You would like to be sending kind of parallel signals both to the locomotor muscles and the respiratory muscles because as part of the oxygen transport cascade, we would like to increase ventilation in proportion to exercise intensity to make sure that we can facilitate gas exchange. So this central command has been really implicated heavily as the primary driver or factor contributing to that phase 1 response to exercise. Because it is very, very fast and it doesn't rely on feedback related mechanisms and it appears to be intensity dependent. Okay? So, if we start looking at, and you can see in the cutoff titles of each slide, I'm basically providing evidence in whether or not the slide provides evidence for or against something, okay, and the actual mechanistic pathway. So when you're studying, you know that you should have an appreciation for the midterm that the questions come basically right off the slide. So if you pay attention to some of these little cues, understand the experiment, what was done, basically what was excluded, what was included. You know, the slides are really, really there. There's no tricks. I'm not going to ask you about the name of my dog or anything like that. All the information's on the slides. So it's just a matter of the cues are there in the titles as well. So this is a really cool experiment. This was actually done by a colleague of mine from the University of Toronto, this guy Jim Duffin. And Jim, I think, actually is the guy that probably put me on the path to be here. I was a terrible graduate student. I showed up from Brock University wanting to be a high school teacher, realized I didn't have it in me to be a high school teacher. I just didn't really want to babysit people, but I wanted to be in an educational environment. So I went to Queens, the only grad school I got into. It's true, I went to Queens, and I don't know, I really didn't do very well for about the first 14 months or so. I just was out kind of partying and drinking and playing basketball and tennis and doing whatever I was doing. And and then all of a sudden I was like, man, I gotta get my thesis done. So I took a trip down to Toronto because this guy had this really cool kind of measurement system, this rebreathing system. And I got down there and you'll meet somebody hopefully in the period of your careers or your lives where somebody just really inspires you. And at the time I wasn't particularly inspired by my supervisor, but this guy and his postdoc just gave me the whole day and explained everything to me and spent all this time with me, and they were, I don't know, just incredibly generous with their time and their knowledge. And I went back, and that day, once I got back to Kingston, it was over for me. And then I knocked out my thesis in 6 months, and then I transitioned into a PhD and got my job here. And it's interesting, so I like these types of experiments because this person obviously has an important meaning to me. But this is a pretty cool study that they did that shows you don't need to, take the cortex out of an animal and cut its nerves and stuff like that and stimulate its diencephalon to get some kind of cool information about the control of breathing during exercise. So what I'm showing you here is ventilation on time, and I'm showing you here in the inset muscle EMG activity on time. So this was a 3-minute bout of exercise. So basically what they did for this was to look at the role of type 3 muscle mechanoreceptors is that they basically created a leg kicking device, a machine, where they put healthy people onto a table like this, and they fix their leg into a device that would move their leg basically through extension and flexion at a given kicking frequency. Pretty simple. So in this case, the subject is not actually activating their quadricep to generate the contraction. The machine is doing it. They're told to relax and not resist or work with the device. So and then because the machine is doing the activity, the idea is like if you're not thinking about exercise, there's not much in the way of voluntary activation of the muscles. So the idea is that there's not much in the way of central command. If you're not actually activating the muscle and it's being moved just through space, there's not any metabolic stress, right? Because the machine is doing all the metabolic loading for you. So what you're left with through basically a process of deduction is just activation of the proprioceptors and the mechanoreceptors, just through movement of the limb, the joints, the tendons, changes in blood vessels with muscle pump effect, things like that. And so what they found is that when they did this, as soon as they started to passively kick the leg, ventilation increased in a very rapid manner. So the idea is no central command, no metaboreceptor feedback, no central or peripheral chemoreceptor feedback, and presumably no central command feedback because they were asked to relax and not work with or against the kicking device. And indeed, it's a nice little part of the experiment. They included muscle EMG activity in there to show that there was no actual activation of the muscle. So muscle EMG activity didn't increase. So the muscle was moving, but it wasn't actively moving. If it was actively moving, you would get bursts of the quadriceps EMG. But it wasn't happening. So these are muscles that are quiet, not voluntarily activated, not metabolically active, just moving through space passively. And with passive movement of the leg, you get an immediate increase in ventilation that begins to wane off a little bit, but nevertheless, it certainly stays above resting values. And so the idea here is that stimulation of type 3 mechanosensitive afferent nerves through movement and distortion or distension of blood vessels, muscles, joints, and tendons has the potential to contribute to that immediate increase in ventilation from the rest to exercise transition. And this is despite the fact that there were presumably no changes in central command as indicated by the lack of change in muscle EMG activity or muscle metabolic demand or disturbance. So, kind of a cool experiment where you just have a machine do the activity and you don't necessarily need animal studies to do kind of reductionist studies. There's still some nice integrative human physiology that's being done. So, this is some experimental evidence for mechanoreceptors. So, the same group does a really clever extension of this experiment. So, to orient you here, so here what we have is we got a period of rest, we got a period of passive leg kicking, we got a period here of active leg kicking, and then we got a period here of recovery to rest. So in the upper panel here, I'm showing you ventilation, the ventilatory changes. Here I'm showing you the root mean square of the quadriceps EMG as a measurement of central command, or actual muscle activation of the muscle. And here I'm showing you leg movement frequency. So it's zero at it's silent at rest, it's silent in recovery, but importantly, between the passive and the active leg kicking, this is really, really cool. They kept the kicking frequency the same. So what do you think that means about the level of activation moving from the active to the passive in terms of the mechanoreceptor stimulation? It's going to be the same. Such a cool experiment. You don't need arterial lines or nerve sticks or anything. It's just a cool experiment. You measure ventilation, you stick an EMG electrode on the quadricep, and you just design the experiment to have cool insight. So let's just keep the kicking frequency the same between when it's moving passively and actively. And presumably that's going to result in the same mechanoreceptor stimulation. Right? So it's a really nice experiment in this regard. So what you can see is that when you move from rest to the passive leg kicking, you get an immediate increase in ventilation like I showed you in the previous slide. So the leg kicking goes up to 75 cycles a minute. You can see that the quadricep EMG is effectively silent, so there's no central command, but nevertheless ventilation increases. Further evidence for a role of mechanoreceptor stimulation of ventilation during exercise. Now, at this time point here, they ask the participant in basically, for example, in 3, 2, 1, the kicker's going to turn off. Now maintain the same kicking frequency at 75 cycles a minute. So now they go from passive to active leg kicking. So now you can see immediately they activate their quadricep to maintain the same leg movement frequency, and look what happens. When you add on top of central command and probably maybe some mild metabolic disturbance of the muscle, on top of what is presumably a constant type 3 mechanoreceptor activation, you get a further increase in ventilation. So the effects are additive. You have some contribution of the mechanoreceptors, but then you have an added contribution of central command. So you can see that moving from one to the other, mechanoreceptor stimulation to mechanoreceptor plus central command increases ventilation again. And then once you turn everything off, you remove central command, you remove muscle activity, you can see that ventilation then begins to decline back towards the resting values. So, again, this is evidence for both central command and mechanoreceptor activation contributing to the control of ventilation in somewhat of an additive or synergistic manner. So these things don't operate necessarily in isolation. They're obviously operating in an integrated manner altogether. And what I don't have evidence for is that right now is that if you knock one thing down, does another one upregulate? You know, is there enough kind of plasticity in the system such that, all right, well, I have, a very hyperactive metabol reflex or mechanoreflex, do I require less central command? I don't necessarily know what the answer is. What I do know is that there's an incredible amount of redundancy in the ventilatory control system. And so there's some evidence that if you have active pathways, like you have active or inactive pathways, the control of ventilation seems to be remarkably preserved in the presence or absence of some of these pathways. Okay? So, point is that there's redundancy in the system. So, if we start looking at the role of central chemoreceptors, so now these are chemoreceptors again that are located in the brain that are primarily responding to increases in cerebrospinal fluid hydrogen ion levels. So not only systemic but primarily brain or central acidosis. Okay? So there's a pretty cool genetic model of central chemoreceptor, central chemoreceptor control of breathing, and it's a group of individuals that are born with a genetic mutation in the FOX2B gene. They're individuals that they call, the condition is called central congenital hypoventilation syndrome. So they're basically born at birth without functioning central chemoreceptors. So these are individuals that can hold their breath almost indefinitely. So they become an interesting model to try to look at the role of central chemoreceptors and carbon dioxide and hydrogen ion sensing in the control of ventilation. Basically the premise is that if you exercise somebody without functional central chemoreceptors compared to with central chemoreceptors, if the ventilatory response to exercise, light, moderate, or intense, was the same with compared to without functioning chemoreceptors, then they're probably not all that important. If the ventilatory response to exercise in somebody lacking functional central chemoreceptors was blunted relative to those that have them, that probably means that they are contributing to the control of ventilation during exercise. Okay, so in their absence, if nothing's different compared to when they're in their presence, they're probably not all that important, is the general idea. If there are marked differences, then they probably are. So, these are just examples of the ventilatory response and the end-tidal CO2 responses from an individual control subject and a patient with central congenital hyperventilation syndrome. And so the idea is that when you averaged all the data across all the control and the healthy participants, remarkably, the ventilatory response, the phase 1, 2, and 3 response to exercise were remarkably consistent between those with and without functioning chemoreceptors. Central chemoreceptors. So the idea here is that central chemoreceptors, based on these evidence, do not likely contribute to the control of ventilation during exercise in healthy adults. Because in their absence, ventilation seems to be quite well regulated. The interesting thing about this patient population is that at rest and during exercise, they seem to breathe fine. As soon as they go to sleep, they need to be ventilated. Because many of them will become so asphyxic and apneic that they just don't breathe during sleep. And it's because right now when you're awake and the fact that I'm talking to you and there's stimuli and noises that people don't understand, whatever it is like there's stimuli to breathe. So there's this concept of what we call just the wakefulness drive. Just the fact that you're awake, you'll breathe. And amazingly, that seems to be appropriate to kind of maintain gas exchange. I don't necessarily know how, I don't know what the signal is, but it seems to be there. But when you fall asleep, you withdraw that wakefulness drive. It's called the wakefulness drive because it's present when you're awake. When you're asleep, the wakefulness drive disappears and you become completely reliant on feedback control. So central and peripheral chemoreceptors. So the idea is that when you're awake, wakefulness drives can probably take over much of the ventilation, much like that redundancy in the system that I'm talking about. But when you're asleep and you don't have that mechanism in place, the chemoreceptor control becomes very, very important. And now these patients are very susceptible to gas exchange impairment and severe hypoventilation and even suffocation. So they need to be supported in their ventilation that's during sleep. So a pretty cool, they're very rare, but a pretty cool kind of genetic model to study ventilatory control during exercise and during sleep and at rest. So this is experimental evidence against a role of these central chemoreceptors. So if we look for evidence against peripheral chemoreceptors, now the key here is that I'm going to show you a series of slides, measurements that are done below and above the ventilatory or the anaerobic threshold. Okay? So if you look at the ventilatory response to constant load cycle exercise performed during mild and moderate intensity exercise, which is below the ventilatory anaerobic threshold, when you're getting a systemic metabolic acidosis, right, a disturbance in your blood gases, you're not getting that rise in hydrogen ion levels or a fall in pH in the arterial blood. Okay, so your ventilation is able to match the metabolic demands. You're not really pushing into, driving the ATP provision through, heavy rates of glycolytic flux, and so you don't have much in the way of muscle metabolic and then systemic disturbances and acid-base balance. And so under these conditions, if you compare the ventilatory response and the rest to exercise transition in a normal subject versus subjects from many years ago where part of a treatment for asthma was actually to remove the carotid bodies. So these are individuals who have had their carotid chemoreceptors surgically removed for therapeutic reasons. And you can see that during exercise below the ventilatory threshold, the phase 1, 2, and 3 responses are remarkably similar between those with versus without peripheral or carotid chemoreceptors. So the take-home message here is that, based on this experimental evidence, the peripheral or carotid chemoreceptors do not likely contribute to the control of ventilation, at least not during light to moderate intensity exercise, when there's not much of a way of an arterial blood disturbance in pH or oxygen levels. There's no systemic hypoxia, and there's no real systemic acidosis that would stimulate these chemoreceptors to drive ventilation. Okay? But what happens during heavy intensity exercise if we now exercise at an intensity above the ventilatory or anaerobic or lactic acid threshold, where there is a systemic disturbance in arterial blood gases? There is an increase in arterial blood hydrogen ion levels, and therefore a stimulus to these carotid chemoreceptors to activate, send sensory afferent information to the brain to try to compensate and increase ventilation. And so again, normal subjects and individuals whose carotid bodies have been surgically removed, rest to exercise transition, you see that the magnitude of these phase 1 is again intensity dependent. You can see that it was only an increase to 10 liters a minute. Here it's an increase to 40 liters a minute, kind of demonstrating that the magnitude of the exercise stress was much higher in this particular step. But you can see that in the normal subjects where the phase 2 and phase 3 response don't achieve a steady state and ventilation keeps rising with exercise, look at the hypoventilation that occurs in those individuals without carotid bodies. So they have the same phase 1 response which again is arguing against the role of the peripheral chemoreceptors for that immediate step increase in ventilation. But once you start getting some metabolic disturbance, as exercise time goes on, and now you're getting this systemic acidosis as a stimulus to the carotid chemoreceptors, you can actually see that the ventilatory response is dramatically blunted in the absence of peripheral chemoreceptors. So they have a relative hypoventilation. So the take-home message here is that when the carotid chemoreceptors are intact, they are likely contributing to the phase 2 and phase 3 response during heavy intensity exercise, above the ventilatory or anaerobic or lactic acid threshold. Because in their absence, ventilation is not normal. So the phase 2 and 3 response to exercise above the T-vent is attenuated when the carotid bodies have been surgically removed. And this suggests that peripheral chemoreceptors likely are contributing to the control of breathing, and this is key during heavy intensity exercise. So question on the exam. Carotid chemoreceptors, peripheral chemoreceptors contribute to the control of breathing during light and moderate intensity exercise. True or false? False. Right? So, this is so weird that they did this. I don't really get it. I don't have luck with this route. So, if you look now at incremental exercise, so now this is, again, a very similar circumstance where we're looking at exercise-induced increases in minute ventilation, and on the on the x-axis, we're looking at exercise intensity as a percentage of maximum VO2. So this is now a graded exercise test, going from light into moderate and moderate into heavy and maximum exercise. And so, again, just like in the previous experimental condition, you have the healthy normal control subjects with intact carotid chemoreceptors, and then you have these carotid body resected individuals. And so at this particular point here, this point marks the ventilatory, the anaerobic threshold, the intensity of exercise that typically occurs at, which in this case, as I think I said before, was somewhere kind of in the 50 to 70% of peak VO2 range. And you can see that just like in the slide that I showed you before, during light and moderate intensity exercise below the ventilatory or anaerobic threshold, the ventilatory response is remarkably similar between individuals with compared to without intact carotid chemoreceptors. But when exercise intensity increases and you're getting this systemic acidosis that's occurring and you're getting these blood-borne metabolites that are there to activate the carotid chemoreceptors, you can see that there's a dramatic blunting of ventilation. So you can see here in this case you're looking at about 85 liters a minute versus about 55 liters a minute. So during very heavy intensity exercise, when there's a considerable metabolic stress, the carotid chemoreceptors are contributing importantly to the control of ventilation. Most likely through that feedback sensing related mechanism of the carotid chemoreceptors that are sensing that hydrogen ion that we know is increasing. And remember I showed you the slide in last class where I showed you it was the Wassermann plot where it was ventilation on hydrogen ion and ventilation against PCO2, and the relationship between ventilation and hydrogen ion was quite a bit linear, but then ventilation and PCO2 rose early in exercise but then decreased. So we had the ventilatory control of arterial carbon dioxide levels as a means of regulating arterial hydrogen ion levels. And so this is presumably one mechanism responsible for that, of driving ventilation in proportion to the acidosis of the blood. And so again, this is evidence that ventilation below the anaerobic threshold is not likely being driven, at least not importantly, by the carotid chemoreceptors, but above it is. So, we've talked now about central command, mechanoreceptors, central and peripheral chemoreceptors. We're left now with metaboreceptors. So, within the muscle, receptors that can respond to metabolic disturbances due to muscle activation. So the first thing that we need to do is we need to have an appreciation that metabolic disturbances of the muscle first can activate a type 4 metaboreceptor. That's the first thing, because if metabolic stress doesn't activate a metaboreceptor, then it's probably not a factor that's going to send sensory afferent information in relation to metabolic stress. So in this particular experiment, these are studies that were done by Mark Kaufman's group in Penn State, I believe, years ago, about 30 years ago, where they stuck a needle and they identified type 4 nerves. So these are direct recordings from type 4 nerve afferents in the muscles of animals. And what they did was that they induced a 60-second static contraction. So this is like a 60-second tetanic contraction that's electrically induced. Okay, so they electrically stimulate the motor nerve. The muscle then basically contracts for 60 seconds at a very high intensity. And for those of you that kind of done that ischemic hand grip, you know that if you occlude the blood flow and you're activating the muscle, it'll be metabolically disturbed. Okay? So they do a 60-second static contraction of the muscle, and they see that there is a contraction-dependent increase in type 4 afferent nerve activity. So, stress the muscle with a contraction, the nerve fires. And these are those metabo-sensitive nerves. But now, again, if you want to know whether or not that's truly due to a metabolic disturbance, then mimic the metabolic disturbance, but in the absence of an actual contraction. So what they did in this case is that they take this, they still record the nerve, but now what they do is now they inject, they 15 seconds before the measurement, they do an injection of lactic acid into their arterial blood, but with no muscle contraction. So they mimic the metabolic stress of the muscle, but without the actual muscular contraction. And again, when you get lactic acid, which presumably is dissociating into a hydrogen ion and a lactate ion, you get a very similar increase. Once that metabolite hits the muscle, you get a very similar increase in type 4 afferent activity. So the idea here is that both static muscle contraction and interarterial injection of lactic acid, which simulates muscle contraction and fatigue, increases type 4 muscle metaboreceptor activity. So the idea here is that during heavy intensity exercise, when there is a local muscle metabolic stress, muscle metaboreceptors have the potential. It doesn't say that they do, because we didn't measure phrenic nerve output, we didn't measure anything. It's just saying that this is providing the proof of concept that these nerves can fire in response to a metabolic stress and send information to try to signal ventilation. So you can imagine what the next experiment is. Do something very similar, but measure phrenic nerve activity. Right? So indeed, that's what they did in subsequent studies. So here what you have is you got muscle tension development. So again, they do a 60-second period of a static tetanic contraction electrically stimulated with simultaneous measurement of phrenic nerve activity and muscle pH. So now they're looking at, during static contraction, they're looking at the level of muscle acidosis and the corresponding rise in phrenic nerve activity. In this comparator experiment, they inject hydrochloric acid into the arterial blood, but again, with no muscle contraction. So now, no actual contraction of the muscle, but you've mimicked the metabolic state of the muscle. You've created an intramuscular acidosis by infusing hydrochloric acid. And so what you can see is that in the setting of actual muscle contraction, as muscle pH declines, you get a time-dependent and pH-dependent rise in phrenic nerve activity. So in the previous slide, we showed that those type 4 afferents will respond to metabolic stress. And now, in the setting of metabolic stress, presumably when those type 4 afferents are being activated, you are getting increases in the neural equivalent of ventilation. Now, in the absence of muscle contraction, so now here, there's no proprioceptive information from, say, type 4, from the type, what would the type 4, the type 3 mechanoreceptors. And so when you induce an intramuscular acidosis, you get similar increases in phrenic nerve activity. So the analysis or the interpretation of this is that in the setting of muscle metabolic stress, activation of type 4 muscle metaboreceptors can contribute to the control of ventilation. Now, translating this into the human model of exercise, it really probably depends on the fact that this is exercise intensity dependent. So if there's not much of a way in metabolic stress at the, in the muscle itself, this mechanism of driving ventilation is not particularly active. But as the metabolic stress of the muscle increases with increasing exercise intensity, you would get an intensity dependent increase in its contribution. And so during heavy intensity exercise in humans, presumably, type 4 sensory afferents may relay sensory information about the metabolic state of the muscle to the respiratory control center, which would signal a compensatory feedback-related increase in ventilation. So again, pretty reductionist from animal trying to translate into humans. So I think I've said this before. I think one of the most influential exercise physiologists that's kind of been around the last 50 years is this guy Jerry Dempsey from the University of Wisconsin. And he's done these pretty cool studies with this guy Marcus Amen. I think I showed this, or you got shown this in the cardiovascular section, where they do intrathecal injection of an opiate, so fentanyl. So basically what they do is that in these studies, they will inject a needle into the intrathecal space, so basically into the spinal column, and what they do is that, so this is not an epidural anesthesia, this is into the intrathecal space, and then so what they end up doing is that they infuse a mu-opioid receptor, a drug, so an antagonist drug, that basically blocks migration of sensory feedback information from anywhere below the block. Okay, so in other words, when they put the intrathecal fentanyl into this part of the spine, what ends up happening is that you're not getting any sensory feedback information from the lower limbs, for the type 3 and type 4 afferents. At the very least, they're blunted to a dramatic extent. Okay? So in this experiment,